Today we discuss about the complement system that comes under the serology and immunology topics. So first we discuss about that what is the complement system and who discovered it, what is the history of the complement system. So first we discuss about the history. Actually, complement system discovered in 1894 by the Bordet scientist. It represents lytic activity of the fresh serum. Which activity it represents? It represents lytic activity of the serum. Actually, there are three types of complement system. That is, first is classical complement system, second is alternative, and third is mannose binding lactin type complement system. In complement system, what happened? Actually, in complement system, they phagocytose or lyses the pathogen that enter in our body with one series of the one type of series of a chemical reaction by which different types of complement protein will be activated, and then phagocytosis or opsonization will be occur of that pathogen in this process. What? Opsonization. What happened in this? In this, a series of complement protein will be activated. Series of complement protein will be activated in this process. Okay, it represents lytic activity of serum, and its lytic activity destroyed when heated at 56 degrees Celsius for 30 minutes. Because it's a high temperature, when we heat at a high temperature, the pathogen will be destroyed at that temperature. So this is the history of the complement system. Next, what is the function of the complement system? The main function of the complement system is the opsonization to enhance phagocytosis. Already I discussed in previous slide. The main function of the complement system is the opsonization. What? Opsonization to enhance phagocytosis. Another, phagocyte attraction and activation. What happened in this? When series of complement protein will be activated. Series of complement protein. When this protein will be activated, they lyse the pathogen. What? They lyse the pathogen or infected cell. Regulation of antibody responses. They also regulate the antibody response. Clearance of immune complexes. Clearance of apoptotic cell. The cell which are apoptotic in nature, by the complement system, they remove from the body. What? Remove from the body with the help of complement system. Next, protein of the complement system. What are the protein of the complement system? Actually there are several protein in complement system. In the sequence of the protein by which activated are different in different pathway. But there are the several number of complement protein. The protein will be indicated by like C1, C2, C3, This is the name of the protein. The number is indicated the sequence by which they are discovered. The number what indicated? Number indicate in which sequence they are discovered. Means C1 is discovered first, after that C2, after that C3, like that. Okay. Another protein is factor B, D, H and I. These factor B, D, H and I. This protein or factor, actually there are three types of complement system, right? First is classical, right? Second is alternative, and third is mannose binding protein. Mannose binding protein. Okay. These are the three types of complement system pathway. So this C1 is in classical pathway, this is alternative pathway, and this is mannose binding pathway, helping mannose binding pathway. So mannose binding lactin, MBL associated serine proteases, this all are help where? In third pathway, that is mannose binding pathway. And last one is C1 inhibitor, C4 binding protein, decay accelerating factor, complement receptor and protein S. What? C1 inhibitor, C4 binding protein, decay accelerating factor, complement receptor. Now what is the definition? What is the definition of the complement, and what is the activation of the C protein? How activated? So C activation, alternation of C protein such that they interact with the next component. How the C protein will be activated? They activated by a alternation of C protein such that interact with the next component. Next is C fixation, utilization of C by antigen antibody complex. When C protein will be fixed, then they are utilized by as an antigen antibody complex. Next is hemolytic units, dilution of serum which lyses 50% of standardized super suspension of antibody coated RBC. C inactivation, denaturation of an early C component resulting in loss of hemolytic activity. Clear? And last one is convertase and esterase. Altered C protein which act as a proteolytic, they act as a C component. They act as C component. Now activation products of complement protein. Activated component are usually overlined. Already know they are C1, C2, C3. When they react, they converted into a first of all they are inactivated C1, remember that thing. When they activated like this, a barcode is or line is present. This means they are activated. The activated components are usually overlined. Example C1QRS means they are activated. Clear? When enzymatically cleaved, the larger moiety binds to the activation complex or membrane and smaller peptide is released in the microenvironment. Remember one thing, when suppose this C1, when they activated, they converted into a two fragment like C1A and C1B. Okay, C1A and C1B. B is a larger fragment. What? B is a larger fragment and A is a smaller fragment. This larger fragment bind to activation complex. Activation complex. Whereas smaller fragment released in the microenvironment. Means there is no use of a smaller fragment and there is use of this larger fragment. Okay, remember this. A is a smaller fragment and B is a larger fragment. But there is one exception, that is C2. What? C2. For only C2. That in C2, B is the smaller fragment and A is the larger fragment. Remember, this is the exception only. What? This is the exception. When enzymatically cleaved, the larger moiety binds to the activation complex or membrane and the smaller peptide is released in the microenvironment. Already I told you is visually added to the larger membrane binding and peptide and A to the smaller peptide. But there is an exception, that is C2. In C2, the larger membrane binding moiety is C2A and the smaller one is a C2B. This is only for the C2. Remember this. This is the exception. So now, what is the pathway? What is the pathway of the complement activation? Already I told you that there are three pathways of the complement activation. First is classical pathway, second is Mannose binding pathway, and third is alternative pathway. In classical pathway is the antibody dependent pathway. What? Antibody dependent pathway, classical pathway. And lactin and alternative pathway is the antibody independent. Remember this one, very important. Which is antibody dependent pathway and which is antibody independent pathway. So classical pathway is the antibody dependent and lactin alternative pathway is antibody independent. After that, when complement system is activated, activation of C3 and generation of C5 convertase. In each and every pathway, there is generation of C5 convertase. Very important. What? Generation of C5 convertase. When C5 convertase activate, they activate the C5 complement protein. They activate the C5 complement protein. After activation of C5 complement protein, this C5 complement protein help in lytic attack pathway. What? Lytic attack pathway. The opsonization of the pathogen by phagocytosis, so that the pathogen or foreign particles that enter our body will destroy. Clear? Which is antibody dependent pathway and which is antibody independent pathway. So now, component of the, now we study about the classical pathway. What is the component of the classical pathway first? So the component of the classical pathway is the C1, right? C2, C3 and C4. Clear? C1 protein, C2 protein, C3 protein, C4 protein. This is the C1 complex. The complex in C1 is in C1 complex which ingredient is present? In C1 complex, calcium ion, C1S, C1R, C1Q, these are all are present. Remember this. Which type of protein or ingredient are present in C1 complex? C1Q, C1R, C1S and calcium ion. So the component required in classical pathway is C1 complex, C2, C3 and C4. Okay. Now, classical pathway generation of C3 convertase. Generation of C3 convertase. How the C3 convertase will be generated so that C3 protein will be activated, and after that C5 convertase will be generated so that C5 will be activated. So now we have first target is the generation of C3 convertase, right? So see, this is the C1 complex, right? This is a C1 complex. When this C1 complex will be activated, they activate the C2, right? C2, in C2, what happened? C2A will be removed and C2B will be present. Sorry. C2 is exception, right? Remember this C2 is exception. So this C2B is removed. C2B is removed and C2A will be remain for the binding. Clear? So first what happened? First C1 complex activate the C4 then C2 then C3B. Clear? So C4B, C2A and 3B is a C5 convertase. What? This is a C5 convertase and it leads into a membrane attack pathway. When this C5 convertase will be activated, they activate the C5. What? C5. Right? And after that, this C5 help in membrane attack pathway. Clear? Clear this one? See, this is the stimulation by antibody, especially IgM and IgG. Because in previous slide, already see that classical pathway is an antibody dependent pathway, right? So, there are 5 subclasses of antibody. So, this pathway is depend on which subclass, right? So, this is a IgM, this depend on the IgM and IgG, remember that. The classical pathway is stimulated by antibody, especially IgM and IgG. What? IgM and IgG. It starts with C1q, a huge protein, 4,10,000 Dalton, composed of 18 peptide. Right? Peptides can associate to form trimer. 6 set of trimer make C1q. See here, this is a 6 set of trimer that make a C1q, right? C1q has a helical stalks and globular. This is the globular head and this is a stalk, right? This is composed of 18 peptide, right? Right? So this is a C1q Hughes protein, right? That help in classical pathway. The head are the carboxy and the tail are the amino end. Means this is the amine group and this head is the carboxyl group. Okay? Clear? Now see what happened here. See, this is the proper pathway of the classical pathway. Okay? How the C5 convertase will be formed. See, this is the C1. Where is the C1q? This is the C1q. In previous slide you see that this is the stalk, this is the globular head and this is amine group and this is the carboxyl group end. So this is a C1 polypeptide. They activate the C4. First activate what? C4 will be activated. When C4 will be activated, they activate C2. When C2 is activated, they activate C3. Right? When C3 will be activated, they form a C5 convertase. When this C5 convertase will be formed, they attack like as a membrane attack. What? Membrane attack. Clear? This is the when the C5 will be activated, they activate the 6, 7 like that, right? When 6, 7 will be activated, see here. When 6, 7 will be activated, they go like that and bind to C9. After that, they phagocytose. When poly C9 will be present, they act as a membrane attack complex. They act as membrane attack complex and they help in lytic cycle. And they destroy the pathogen. Destroy the pathogen. So, biological activities of classical pathway components. Components C2b, C3a, C3b, C4a and C4b. C2b, what is the activity? Prockinine, cleaved by plasmin to yield kinine which result in edema. C3a, anaphylotoxin, can activate basophils and mast cells to degranulate, resulting in increased vascular permeability and contraction of smooth muscle cell, which may leads to anaphylaxis. C3b help in opsonine, activation of phagocytic cell. C4a, anaphylotoxin, help in anaphylotoxin, and C4b help in opsonization. What? Help in opsonization process. Okay. Control of classical pathway components. C1 inhibitor. All components are regulated by C1 inhibitor, that is C11H dissociates C1R and C1S from C1Q. C3A is regulated by C3A inactivator, that is C3A and carboxypeptidase. C3B is regulated by factor H and I. C4A is regulated by C3A, I and H, and C4B is regulated by C4 binding protein. So, C4B is regulated by C4 binding protein. Clear? Now, components of mannose binding lectin pathway. What? Mannose binding lectin pathway. What happened in this pathway? In this pathway, the component is mannose binding lectin is itself component, right? Mannose binding, and this is present on a bacteria. So, in this mannose binding lectin, C2 as in classical, C4 as in classical, and MASP2 and MASP1. This is the component of the mannose binding lectin pathway. Now, mannose binding lectin pathway. First what happened? Mannose binding lectin will be present, right? They activate the MSP1 and MSP2. After that, C4 protein will be activated. They convert it into C4A and C4B. C4B is the larger component, so it is the larger component, so it will remain, and C4A release in the environment. Now C2B is C2 will become, and they cleaved into a C2A and C2B, right? They converted into a C3 convertase. It will lead to generation of C5 convertase, right? When C5 convertase will be generated, they activate the B, P and D, right? So C3, this that is the mannose binding pathway. This is the mannose binding lectin is the mannose binding lectin C4, C2, C3 is the component of the mannose binding pathway. Now, what is the component of the alternative pathway? The component of alternative pathway is the C3, B, P and D, right? In alternative system, C3 is the principal protein of concern, exists in high concentration, contain an unstable internal thioester with slow spontaneous or serum protease mediated cleavage. Clear? By convention, the smaller product is designated A and the larger is B. Already I told that a smaller one is A and larger one is B except C2. Except C2. C3b can bind to bacteria and enhance phagocytosis. These are the alternative pathways. The production of C3b can be catalyzed by C3 convertase. C3b can be catalyzed by C3 convertase. This is the factor B, factor D. When they combine to form the factor, they bind to factor B. This factor bind with C3b, right? This binding of propidine stabilizes C3b and BB, C3b and BB. This is the C3 convertase. What? C3 convertase. what happen in alternative system? When C3 convertase will be activated, they activate the C5 convertase. When C5 convertase will be activated, the C5 convertase activate the C5, and already know that C5 will keep into C5a and C5b like that, and when C5b is unstable and is stabilized by C6. When C6 will be activated, they allow C7 like that 8, and when 9 will be activated, they form a poly C9. When poly C9 will be formed, then they act like a membrane attack complex. In that, they phagocytose the foreign particles and they destroy from the body. Oh, clear? Now the complement pathway. See, this is the combination of all the complement pathway. This is the lactane pathway, this is classical pathway, and this is the alternative pathway. Lactane pathway, MBL is used. In classical pathway, antigen antibody is used, and in alternative pathway, microbial surface is used. So remember this all pathway. Most important in complement system is the pathway. So lactane pathway, mannose binding lactane is used. That is microbial cell wall. In alternative pathway, microbial surface is used and factor D is used, and in classical pathway, antigen antibody complex is used. Okay. So biological effect of C5a. C5a help in adhesion, chemotaxis, vascular wall transmission, mast cell degranulation, and cytokinin production. This is the function of the C5. Now, biological properties of C activation products. C2b prokinin, C3a anaphyloxy, and the biological effect of C2b is edema and regulated by C1nh. So, this already I told you in previous slide. Complement deficiency and disease classical pathway. Pathway C1nh, C1, C2, C4. Disease is hereditary, angioedema, predisposition to SLE, mechanism overproduction of C2b, and obstination of immune complexes help keep them soluble. Deficiency result in increased precipitation in tissue and inflammation. So this is all are the deficiency. What? Deficiency of the disease of complement system in alternative pathway, classical pathway, and lactose binding pathway. Clear? So this all about the complement system. Today we stop here. Hope you understand all three pathway, that is classical pathway, mannose binding pathway, and alternative